The human body operates less like a single organism and more like a highly structured society composed of trillions of individual cells. In the 19th century, physician Rudolf Virchow captured this reality by describing the body as a cell state in which every cell is a citizen. Yet for over a century, traditional pharmacology has governed this vast state by treating its citizens as entirely uniform. A standard chemical dose is prescribed based on statistical averages across millions of people. In reality, these cellular citizens are highly diverse. Each carries a unique metabolic code, specific genetic variances that dictate exactly how fast or slow they process chemical compounds. Applying a uniform chemical intervention to a population with such deep genetic heterogeneity forces a mathematical compromise. It means the standard dose will be toxic for some, ineffective for others, and optimal only for the statistical middle. Pharmacogenomics, or PGX, abandons this trial-and-error approach. It uses a patient's exact genetic makeup to guide their specific drug therapy. The logistical scale of this effort is immense. A single patient carries roughly 3 billion base pairs of raw genetic data. Raw DNA sequences, however, offer no immediate clinical value. To adjust the dose of a single pill, a physician needs a system capable of parsing those 3 billion data points in real time. Building that capability requires a massive infrastructure. It starts with extracting data from population biobanks, structuring that data into biochemical pathways, and finally translating those pathways into standardized dosing rules. Precision medicine relies on this engineered data pipeline to turn microscopic biological variants into safe, macro-level patient care. Before any gene can be reliably linked to a specific drug response, researchers need overwhelming statistical power drawn from a highly diverse human population. Historically, genomic datasets have suffered from severe homogeneity, skewing heavily toward populations of European descent and failing to represent global genetic variants. The National Institutes of Health established the All of Us research program to directly correct this data deficit. This chart shows the geographic distribution of the program's participants. It maps over 830,000 enrolled individuals spread across the United States. This chart shows the demographics of that cohort. Crucially for precision medicine, the top bar reveals a 47.5% non-white participant representation. The program captures multiple layers of data, matching their DNA sequences with electronic health records, physical biosamples, and returning hereditary risk reports. This structured biobank establishes the statistical ground truth researchers need to definitively prove that a specific genetic variant consistently causes a specific clinical outcome. Once the raw population data is gathered, it must be organized into coherent biological logic. Organizations like PharmGKB operate at this layer, performing exhaustive literature reviews to bridge raw genetic associations and clinical applications. They structure this evidence into pathways. First is the pharmacokinetic, or PK pathway, diagramming how specific liver enzymes, like CYP2D6, act on compounds. Pharmacokinetics maps what the body does to the drug. It tracks absorption, distribution, metabolism, and elimination. The complementary model is the pharmacodynamic, or PD pathway, which maps the resulting cellular interactions. Pharmacodynamics maps what the drug does to the body, detailing how the active chemical binds to receptors and triggers a physiological response. These carefully diagrammed pathways serve as the biochemical blueprints. They explain exactly why a genetic mutation in an enzyme leads to a failed or toxic drug response. A complex biochemical blueprint, however, is difficult to interpret during a fast-paced clinical consultation. The Clinical Pharmacogenetics Implementation Consortium, known as CPIC, takes these biological pathways and encodes them into strict clinical rules. CPIC uses a two-step translation process to convert genetic pests into actionable advice. Step 1 maps a raw genetic test result to a predicted clinical state, called a phenotype. Step 2 translates that phenotype into a numerical dosing recommendation. This two-table matrix compresses millions of biological data points into a single binary decision that a doctor can safely execute. The transition to standardized precision medicine does face bureaucratic obstacles. A major point of regulatory friction exists between the FDA's tables of pharmacogenetic associations and CPIC's own clinical implementation guidelines. For example, the two institutions offer differing guidance on adjusting tacrolimus dosing based on the CYP3A5 gene. They also disagree on actionable markers. CPIC provides dosing guidance for the RYR1 gene, while the FDA relies on an entirely different gene. This creates an immediate operational challenge for hospitals. Administrators must choose between competing evidence grading systems to establish their internal prescribing rules. Aligning these institutional standards through unified data governance remains a necessary step to making genotype-guided therapy a universal practice. Once the guidelines are established, they must be physically integrated into the hospital environment. The ultimate delivery vehicle for this data is the electronic health record, or EHR. It is a digital, real-time version of a patient's medical history. Massive integration projects, such as the Mayo-Baylor Write-10K study, are implementing preemptive sequence-based testing. They sequence a patient's DNA and embed the pharmacogenomic profile directly into the digital chart. Historically, physicians relied heavily on a reactive model, adjusting a medication dose only after a patient suffered an adverse reaction. This system replaces that delay with a preemptive alert embedded directly into the prescribing workflow. The true utility of pharmacogenomics is temporal. It intercepts a dangerous standard prescription with a genetic warning before the patient even takes a single pill. This vast interconnected infrastructure of electronic health records and genetic databases forms the true backbone of modern clinical genetics. Massive biobanks map human diversity. Research groups translate those genetics into biochemical pathways. Consortiums encode those pathways into dosing rules, and hospital software fires them as preemptive alerts. This pipeline systematically removes the guesswork from chemical interventions. Rudolf Virchow envisioned the body as a cell state over a century ago. Today, the medical system possesses the architecture required to govern that state with absolute chemical precision. We have moved past statistical averages. In precision medicine, the patient's exact genome